All right, this is directions for how to make a bioassay lab for testing pollution, and does pollution actually affect plant life. For this experiment, I'm looking at cigarette butts, and so I have a whole bunch of cigarette butts here that I stole from a can of cigarette butts that the cooks put in the little canister back behind the gym. So I just kind of stole it without asking. And now I have a nice little container of all these cigarette butts. And I want to see, do these cigarette butts actually prevent seeds from germinating? Are they harmful to plant life when we litter them on the ground? The first thing I've done is I've laid out 6 different beakers here, and each of them have a bag, okay? Test 1 is the control. I have spring water here that I've collected from the natural spring in the area. So this is nice, pure water, and we're going to test to see how seeds sprout in there. Right here, this is gonna be 100% cigarette water in the paper towel that is soaking inside of this bag. Test 3 is gonna be 10%. Test 4 is going to be 1% cigarette. Test 5 is going to be 0.1% cigarette. And test 6 is going to be 0.01% of the cigarette solution. So we're kind of diluting it as we go further along, and we're going to see how that affects seed germination. I'm going to measure out 100 milliliters of the pure water. And I'm going to pour that into my test 1 spring water control so that I have 100 milliliters of that. Before I get my graduated cylinder dirty with the cigarette water, I'm just going to fill up all the other beakers with 90 milliliters of water. Okay, now I have this cigarette smoky water, and believe me, it smells like cigarette smoke. It's really bad. I probably should not have done this in the classroom, because now the classroom might smell like this. I don't know. But you got to measure out 100 milliliters of this. So 2 should only have cigarette water in it. I think I accidentally put some distilled water in there. But we're just going to put cigarette water in number 2. So that is our test 2 100% cigarette solution. And our hypothesis is that as we decrease in cigarette concentration, we should increase in the seed germination. Now I want to create a 10% cigarette solution in number 3. So all I have to do is, I've already got 90 milliliters of spring water, and I just need 10 milliliters of that gross water. We're not going to use all 100 milliliters here. So I'm just going to measure out about 10 milliliters of the cigarette water, and then we can add that to number 3. Okay, so now we have 100% and 10% cigarette solution. To make a 1% cigarette solution for test 4, all I have to do is take 10 milliliters of my 10% and then dilute that with the 90 milliliters of number 4's spring water. I'm going to add 10 milliliters of number 4 into the graduated cylinder, and then I'm going to add those 10 milliliters to number 5. Now this is a 0.1% cigarette solution, but I would hardly know it. You can hardly tell that it is contaminated. That's okay. We're going to make a 0.01% final. So I folded up a paper towel here, and I have my little radish seeds as well that I'm going to place. I'm going to place 10 in each one, but first I have to soak it with my solution. And what I did is I just have a pipette, and I'm just going to kind of drop it throughout the paper towel, kind of squirting it around. Make sure the whole paper towel gets wet, okay? You don't want any part that's not soaked. After it's fully wet, I've put 10 seeds on the front end of the paper towel, and it actually sticks up pretty well. Even if I store this vertically, the seeds aren't going to move. All right, here are our results after 5 days. Obviously, I was looking at the data over every single day, but after 5 days, the radish seeds have really grown very well in the spring water. After just 24 hours, they had all sprouted. and they had shut out roots, and now these leaves are growing. You can see some phototropism happening as well as the light is coming from this direction. If we look over at the cigarette water, obviously there's quite a difference. There's no leaves even present here, okay? There were leaves here, there were no leaves here. Only 1, 2, 3, 4 have actually germinated, and they've not done so very successfully. The other ones haven't even successfully germinated at all. When we go to the 10%, okay, they're doing a little bit better, and some of them seem a little shorter, but they're not enormously different from the spring water. As we get to less and less polluted, you can see that they are still doing just fine. But for sure, the 100% cigarette water, that definitely did have an effect. And you can still kind of see some visual differences between the spring water and the 10% cigarette water. So this experiment was a great experiment. I think something else you could start testing is phototropism, and I don't know how you'd maybe quantify some of that, but something to consider is not just measuring the number of like when the seeds germinate, but also looking for the presence of leaves maybe. Yeah, looking at both those factors might help you create a graph about this.